morning. It's wonderful to be here. Madrid, nice weather. Figure out how it would be in Copenhagen, 2 degrees and snow. So it's wonderful. I'm going to tell you a little bit about our approach to next generation sequencing. And it's very nice that Bruce has made this nice introduction and Walt has told us about the mitochondrial DNA, because then it's much easier for me. So this method is called second generation sequencing also by some people, and massively parallel sequencing, and that's maybe the best word. And it can be used for a lot of things, and I don't need to go through this, all these things now. I would just say that although I come from a classical forensic genetic laboratory, we are actually using next generation sequencing for a lot of problems related to molecular pathology and, for example, genetic heart diseases, but a lot of other things too. It's a very efficient tool, but I will not talk about this today. In Copenhagen, we have these 3 small disk devices. The first one was this 454 technology, and we still use it, although it's slow and it's expensive, because it has the capability to sequence long stretches of STRs, and so that's necessary in some context. We have a MySeq and a ForenSeq, and we use it for various things, and we are trying it out to see where the strengths are and the weaknesses, and we have some ion PDM things. And we are mainly using this for our classical forensic genetic typing, sequencing, and I'll tell you a little bit more about this later. When you're going to work with this for STRs, for example, you have to think about the read lengths. And there are some restrictions presently concerning the read lengths. And if we really want to sequence, for example, a large STR region, then it would be a very good idea to sequence from either primer to primer or from flanking regions to flanking region to be sure that you've got it all. And that may be a challenge if you can only read maybe 100 or 200 bases, because some regions are maybe 300, 400 base pair long. The Roche, GS Junior, and the larger devices can do this. The Illumina MySeq and the chemistry that it comes with has some difficulties, but actually we can sequence a lot of the STRs with the Illumina chemistry and the MySeq. The ion torrent has a number of chemistries that we can use, and one of them is a chemistry that can cover approximately 400 base pairs, and that is enough for the great majority of the STRs. So we actually have a choice for the great majority of the STRs. The method that we use for the 454 STR sequencing is a classical one here, with amplicon sequencing, with multiplex identifiers in the PCR primer sequence, and we use PCR primers to amplify in the first step, because it's very efficient and we're used to this and we know the pitfalls, etc., and it makes the whole process very sensitive. So that's very nice. And we can work it on these things in one reaction, so everything is very nice. And when we come to this question about the region that is going to be sequenced, we could maybe suggest to go from primer to primer, but maybe it's not necessary, and maybe the classical primers that we know from the capillary electrophoresis. But I think it's essential that you include flanking regions for this purpose, so that you are sure that you have sequenced these things correctly. And I also think it's very nice to have the possibility to see the flanking region sequences if necessary. That's a good idea as far as I can see, and we have published our ideas of how to do this, and you can see it here. It's a rather old thing now. And here you see some data which were obtained with the 454, and you can see it's one of these D12 results. And you can see there's an allele here and an allele down here. And then there are some errors. And what is this? Can you guess it? Yes, of course you can guess it. Stutters. We see stutters here because it's a PCR reaction, and it's obvious that there must be some stutters and there must be some errors here and there. This is one of the first things we did. That was made with one of the big flex, 454 flex, and you can here see that we were very happy to see that the results were actually quantitative in the sense that the number of reads for a given allele corresponds to what you would see in the capillary electropherograms as the peak height. So we were very happy. And it's still like that. It works very well. We were happy to see the polymorphism could help us in some cases where we, for example, here had a paternity case with the father, mother, and the child has 3 alleles, and this allele can come from both of the parents, and then we sequenced it, and you can see that this pattern, 30 from the mother, is the same pattern as the 30 with the child, so we can now say that this allele comes from the mother. So it's nice. And here are some other examples on the increased discriminatory power of these sequences. We made a small analysis of 3 very informative loci, and we realized that there was a great increase in the power of discrimination and the paternity exclusion power and the typical PI. So it's really a good tool. And not surprisingly, approximately 30% of the 1 allele types obtained with capillary electrophoresis turn out to be from individuals who were really heterozygous when we sequenced them. And until now, we have seen 8 microvariants. I know that other laboratories may have seen even more, but we have a process now going on in our lab where we want to make databases for all the systems that we want to work with in the future. So in the beginning, we will start with approximately 200 individuals, and then later we will catch up with the results that we get from our usual work in paternity testing, immigration testing, and in crime case work. We work with an experimental kit from Life Technologies with 9 STRs and aminogenin. And I got a lot of experience with this. I'm pressing and pressing. Okay. After the PCR, the primers are digested. That's their technology. And the amplicons have been adjusted to between 75 and 170 base pairs. That's necessary. I'll explain it later. And we sequence it with the smallest chip that is available. And there's a statistical tool which is a plug-in for the analysis of these STR regions, and we use that also. Although I have to admit that we often take the data out of this environment and put the data into our own environment because we want to do the analysis in a slightly different way in some cases at least. The point about adjusting primers for this kind of second duration sequencing, or NGS, is important, at least for the time being. If you look at this, you can see that the distribution of the lengths of these systems and alleles are approximately like this. It will not work with the present technologies in one shot with NGS. You have to do something, and we have to have a more homogeneous size distribution of the alleles down here, or the STRs down in the soup. So that has been made, and that is being done with all these kits, at least presently. Oh, okay. It's going to be a long day. Okay, so we made a pilot experiment here, and it's published, so you can read about it here. So we wanted to see the sensitivity and the concordance with previous results obtained with capillary electrophoresis, and we also wanted to include some of the samples that we knew were difficult, at least when we were going to analyze with capillary electrophoresis. But after all, it's PCR, which is the essential point in the beginning, and therefore we could expect the same difficulties with this methodology. And everything that we're doing for Casework is accredited in Copenhagen, so it's rather robust. And we had also the problem with mixtures into considerations, and of course it's funny to see 1 to 1,000 because it will not be easy to identify 1 in 1,000 with the present technology. But we wanted to do it. And here you see the results as they come out of this plug-in that comes from Applied Biosystems. And we see the same phenomena as usual. These are the alleles, and there will be some stutters, and should be there, and there are some other artifacts. So it's the same picture as you were used to. What about noise? You can see a noise plot here, and it's not worse than what we see with STRs and capillary electrophoresis.
Niels, if you point towards AV,
okay, so that will be. Okay, I'll try to do that. So noise is not a bigger problem here than we're used to. And allele balance, the same as with many of the capillary electrophoresis results. And stutter ratios, again, it's the same, almost as with our usual techniques. And what about dropouts, allele dropouts, and locus dropouts? Well, if we look at the DNA amount here, you can see this is 10 picograms and 100 picograms and 500 picograms. So you can see that it works at least from 200 picograms in our hands. So that's also very promising. And we saw the same results with NGS and with PCR capillary electrophoresis data, so complete concordance. And now we will go to the mixtures. This is a 1-to-1 mixture, so they should be equal. And here we have a 1-to-20 mixture, and the green ones down here represent the minor component. And again, these results are promising, and we are going on with this. We're not going on with the 10-plex because it's a little bit old-fashioned now, so we are doing other things than this. So we went to the challenging material, and here you see some results obtained with capillary electrophoresis, and you can see that there are only results with the short systems, the short STR systems here, when you analyze with capillary electrophoresis, and these primers and they can identify these STRs with that technology. If we go to the NGS, the primers have been moved closer to the repeat regions, and here you can see that there are results with all the systems, all the 9 systems, and a melogenin. So it's again in a significant improvement of the results. Now we have to discuss nomenclature, because we get a lot of more information with the sequencing. And how are we going to handle this? One point is that it's a good idea in the databases to have the complete sequence, but it's very difficult for a human being to find out, okay, what is this? So we would very much like to have some kind of condensation of the results, and we have come up with some suggestions here. Wow. Okay, let's take it like this. We can count the number of repeats, and here you can see that we have counted that this D2 allele has a total of what we will call 18 repeats, but they're different. There's a motif here with 6 TGCC and 12 TTCC, and then there is a mutation here. Is this one? In the flanking region. And the other allele is at 23, with 7 of these, 13 of these, 1 of these, 2 of these, and a mutation again here. So, this is our suggestion for a nomenclature, and we're using it in our publications now, and we hope very soon there will be a common agreement about the nomenclature, and the International Society for Forensic Genetics is working on this. So this is how it would look like if a person's STRs are described in this way for these 9 STRs. So we can summarize all this and say that it works for the most important things that we have worked on until now. And especially the mixtures are promising, I think, and we are actually trying to implement this. It takes a long time to implement this kind of new technologies, and we are trying to follow the same strategies as we have for any other kind of testing in our laboratory, which among other things means that we have more than one device for these things, so we separate it in various groups, etc., etc. Which means that a lot of people have to be trained for working with this, and that we are in this process, and we are building up databases, etc., etc. One of the things that we need is to have better data analysis tools. And I know that the companies are working on it, and we are also working on it so that we can take out the data from the various devices and treat the data as we think they should be treated. And we need simpler, faster, and less hands-on and automation of the laboratory procedures and the data analysis. That's very important because it can take a lot of time to do this. And of course, we need the nomenclature in place. So I'm going to tell you a little bit about the Illumina kits that we tested. We tested this Mix B some months ago. It has a lot of genetic markers, STRs and SNPs, and they are distributed as autosomal and lineage markers, and the purpose is identity and relationship testing and phenotypical traits and ancestry, and it's all in one package. And these data have been analyzed by Illumina, and among others, Copenhagen. We analyzed our own data, and I'll present some of this. And you can see here that the great majority of the results are very nice. This is a question about the coverage, and you can see that some of the markers have a very high coverage, like this autosomal one in here, while others have low coverage. So the balance is not optimal, but it's promising as far as I can see. Here you can see that this one is not balanced comparatively to the other results, but if you take that out, it's fine. And what about the sensitivity? Again, you can see the same pattern as you see when you do capillary electrophoresis. PCR and capillary electrophoresis, and you investigate the balance between the two alleles of a heterozygous individual. Then you hope for having a balance of 1-to-1. This is here. But in real life, you will see a kind of a trumpet so that the balance will be better and better until you reach somewhere between 500 picograms and maybe some nanograms. We also need to think about which markers are actually being investigated with these various kits, and we need to have comparability to the old stuff that we have in our databases and in our files. And you can see here that this concordance is not complete, but hopefully this will be possible to obtain at least some concordance with these new methods. And of course, we would like to see more of these kits with new primers that are closer to the STR repeats, because it will be easier for us to work with these more well-balanced kits. And it would be an advantage for the degraded samples, because the risk of having a break in the synthesis or in the DNA will be lower if the region that you are PCR amplifying is shorter, obviously. And then we hope for improvements of the sequencing chemistries with longer read lengths for the STRs and larger output, and laboratory automation. And for the crime cases, this will obviously be very nice, because we now can get some supplemental information with the SNPs for not only human identification, but also ancestry and phenotypical traits and other things that may be of importance. And the first thing that we will use it for is most likely the difficult paternity cases and relationship testing. And we are actually implementing as good as we can these methods with some of these kits. And again, software tools that are useful. I will not go deeply into SNPs sequencing, but just say that it's much easier to sequence the SNPs, because the regions that are necessary for the PCR are very much shorter, and you can adjust the primers and the sizes of the amplicons, etc., etc. And you can use all the present technologies for these purposes. And we have published work on some of these packages from applied by systems. And you can see it here. A package with 160 SNPs, and can read it in the Forensic Sciences and Natural Genetics. Here you see some images of the coverage and with allele balances, and the results were concordant with the previously obtained results. But of course, there were some of the SNPs which did not work so well. So we found that at least 8 of these SNPs had a large variation in allele balance, and so large that we wouldn't use them in real life. And a lot of small details. And sensitivity, we typed some other groups. Here you see the results from some individuals who were typed with from, let's see if you can see, small amounts. This is 10 picograms. And when you reach these 2 nanograms here, then it will work. And with 500 picograms, it will work reasonably well, but there are some difficulties, of course. Mixtures. Are they coming? Yeah. So we made an experiment with these, with this kit, and ended up with allele proportions here that were rather nice. So here we have the expected mixture proportion, and here we see the observed mixture proportions, and the ideal is this straight line, and it's not a straight line, but it's not bad. The correlation coefficient is 0.98, so this is promising as far as I can see, and it's not worse than the results that we see with PCR and capillary electrophoresis. And here we see some results where we have modified some of the PCR cycles because we wanted to increase the sensitivity of the whole system. And I think that the original recommendation was maybe 17 or 19 PCR cycles, and now we have found that around 25, 27 PCR cycles can optimize the system, but that is the maximum. And here we used it for a difficult sample with 27 PCR cycles here, and you can see the results were nice. And again, it's a little bit difficult to compare all these kits because they have different SNPs, and I hope that we can settle on some standard SNPs for these purposes so that we can obtain comparable results. And we can easily use this for Ancestry, and I have a few slides here. Here we have some results from SNP typing with another technology, and to the right there are some results of applied by systems typing with two different set of SNPs. And here's another sample. And to the left, we have marked the Y chromosome markers, and to the right, we have marked the mitochondrial DNA markers. So these are the results that we can expect in the future. Here we have Frederick. This is how he looked when he was a child, and that's how he looks today. So now we wanted to see what are the results from SNP typing for phenotypical traits, hair color, eye color. So it seems as if he is predicted to be of German, Polish ancestry, and you can see that the hair color is predicted to be blonde, 69%, brown, 27%, and this was in its use and now. And eye color, 94% blue, and most likely you can see that he has blue eye colors. And here you see an analysis of the ancestry, European ancestry. I have some problems with this. Now I will just come up with a warning. We have to be, 1 minute. We have to be critical, and we have to go through all the usual cautions when we introduce new technologies. So that is the most important thing now. And if I can get to this slide, I can tell you that we have a lot of academics who are working on this, and here are the main contributors. And that was all, folks. Thank you.